Welcome everyone to this today's demo. So we are very excited to present you our newest software called Giotto. And just quickly introduce ourselves. So my name is Guocheng Yuan, also known as GC. So I'm a faculty of computational biology at Dana-Farber and Harvard Medical School. And I think I moved this. So this is our team. These are the current members over here, and you will see some of these members are presented later today. Ruben, Chen, Ray, and Hui Peng. And this is our website. And if you want to know more about our lab, what we do, then please check this website. Sorry. What's going on here? So just to briefly introduce what we do. So our lab works in the interface between this biology, technology, and the computation. So in this biology domain, our main focus is in the cancer development. The particular questions we're asking are the understanding the cellular heterogeneity and the mechanism for the gene regulations. And we also interested in development methods for a number of the genomics technologies, and mostly focusing on the epigenomics technologies and the single cell technologies. And for that, we develop a number of the softwares, some of them are listed over here. All right, so just to go back to these today's main topic. So today we're going to talk about spatial transatomics. Just to put things into the context, we all know that each of the tissues is composed of a large number of the cell types, and each of the cells actually interact with each other and with the environment. So recently there's a lot of effort trying to dissect the cellular heterogeneity inside the tissue. Most common ones you must know is the single cell RNA-seq. What it does is that it isolates the cells from the tissue and sequence the transatomic in each individual cells. And that's very important for understanding this composition of the cell types and the cell states inside a tissue. But in the meantime, it loses the spatial information. Right? The alternative approach is the spatial transatomics approaches, which developed more recently. And that approach oftentimes requires the imaging, and sometimes it can be combined with the sequencing technologies as well. But all in all, what it does is that provides not only the transatomic information, but also provides the in situ environment information, which is critical for understanding these cell-cell interactions. So a few years ago, we started to work in this domain in collaboration with Long Kai's lab in Caltech. And we first developed a few computational methods to analyze the seq-fish data that Long Kai's lab developed, methods for the cell type identification and for the spatial domain identification. As you can see, these are complementary informations. Sorry. And then later on, we extended our approach to genome-wide using this seq-fish plus technology developed by Long's lab. So in the past few years, there's a tremendous development in this spatial transcriptomics field. As you can see, there's a lot of diverse technologies that have been developed. And also, there's a huge interest from the biology field. Some of them are listed over here. So together, it becomes a very powerful tool set to dissect the biology and the medicine. Anytime we feel that there's a lack of the computational methods to analyze this kind of data, which motivate us to develop this tool we call this Giotto. So in order to make this toolbox as generalizable as possible and make it as easy as possible so that everybody every biologist can use it without much computational knowledge. So we designed this generalizable and also end-to-end pipeline called the Giotto. So the basic input here is very simple. It's just the cell coordinates as well as the cell by gene matrix. And this can be inferred from almost any technologies that you have. And from that, we carried a large number of the algorithms and the functionalities to analyze the data. Not just the methods we develop themselves, but also we feel that in order to benefit the whole community, we also need to incorporate the state-of-art the technologies that develop by other groups as well. So from that point, we welcome collaborations who are proposing to implement new tools that you guys are developing. So of course, we also welcome collaborations from the biologists as well. So finally, we have this toolbox that can visualize the data. So the details of these toolbox can be found in this preprint we posted at the end of May. And also in this website. The top one is the main website, and these bottom two are the developmental websites with the up-to-date information. Above, and also the developmental links over here as well. So on the top of the menu bar, you can find a number of tabs. For example, you can see this documentations. And if you browse it in, you will see these instructions of various components of the Giotto. And if you scroll down, you will see some galleries that actually generated by the Giotto toolbox. If you go to this installation, it will give you instruction about how to install this toolbox, and I hope you find it very easy. If not, you can let us know. One thing that's quite interesting, I think, to most of you guys is that we also present a number of the examples. This is basically a step-by-step instructions how we analyze the various different data sets. And if you have a similar data sets, you can see how to use that also. So okay, so the if we go back to this developmental website, let's see if we can get there. So the first part is the Giotto analyzer. Right? So if you click on that, all right. So this will give you much more detailed information about the analyzer components, right? So you can also see, the very detailed instructions on the bottom and the examples over here as well. Okay. So if you click on this documentation, it's give you much more detailed instructions on how the method works. And the data sets, you have these data sets that's available. You can welcome to browse. And you have a FAQ session. And they have a new section. Actually, we just released a new version just a couple of weeks ago. So we are very excited about this new news. So compared to the previous one, we have significantly expanded its functionalities. Right? So this is Giotto Analyzer. So if you go back to this main website, we also have, oops, so what is going on here? So website. So what it does is shows these instructions of the viewer, and at the bottom they will show you some tutorials and how to run this viewer. So let me just check something else. Now I talk about the goal of the today. All right. So I hope that at the end of this presentation, you will get these take homes. So first of all, we will have an overview of this pipeline, and then we will describe some of the major functionalities of the Giotto. So Giotto actually has hundreds of the functionalities. We cannot go into detail every single one of them. We only have time to discuss the main ones. And we also we demonstrate how the Giotto can be adapted to address the platform-specific needs. And then the last, I want to show you demonstrate the utility of the Giotto using the various data sets. So, okay, so, and then the agenda for today, we will first start with a number of the talks by the team members about these specific topics. And then I'll just mention a little bit of the logistics. So first of all, the questions can be submitted by the chat room using at any time. And then in the end, we have a separate Q&A sessions. And in that time, you can use the raise hand features. And you can, later on, you can have these presentations. We post this online. Okay. So with that, I would stop and turn it over to Ruben.
So I'm just going to take a few seconds. So GC, can you let the people in that are waiting in the meeting room? I think there are a number of people waiting.
Okay, I'm doing that. Yeah, sorry.
Right. So I'm just going to give a quick introduction. So for everybody who's new, you haven't missed really a lot, because I'm first going to give an introduction about how to start with Giotto and what you need to know just to start with Giotto. So I'm going to quickly go over how to install Giotto and how does a typical Giotto analysis looks like. So there are two different parts. There are two different modules about Giotto. So one of them is Giotto Analyzer, which is designed to do data analysis and to visualize your data. And the other one, and that is actually just a simple R package. And then the other one is the Giotto Viewer, which is an interactive visualization tool. And that is, you can actually run that as a local application in your browser, and you can interactively visualize to everything that you found with Giotto Analyzer. So Giotto Analyzer, as I said, is an R package. Currently, it's hosted on GitHub, and it can easily be installed, which is like install GitHub and so on. We're also working really hard on trying to submit it on CRAN, and once it's there, you can easily install it with, say, just install packages and then Giotto. So although it's an R package, it also depends on some Python modules, but they can easily and automatically install directly when you run Giotto for the first time. Alternatively, if you do not want to install it automatically, you can also do a manual installation, and there's more information on the Giotto Analyzer website. Right. So I think each one of you probably have your favorite tissue and maybe also your favorite platform you want to use to generate some kind of like spatial analysis data set, right? So, and what you all want to do is you want to generate some kind of like insight from the spatial data. And to do that, what is really necessary with spatial information is that you also want to be able to visualize it, right? So we were thinking about how can we facilitate this for everyone and to make it as easy as possible. And so to do that, we were thinking about, we should create some kind of like a very simple program or toolbox that can be run on a local machine and everybody can run. So this is how Giotto was born. And so Giotto really works on all the it's designed to do all the downstream analysis of spatial data sets. Not so much to do the pre-processing of the different spatial data sets, which can be very different. And the reason they're so different is because there are really a lot of different technology that came out in the last couple of years. So this is just a very recent review of the Lundberg lab, where you can see all this different spatial technology that developed, and each of them are really very different. And now even like there's a new technology called PPC. But Giotto can actually be applied on many of these different data sets, as we have already shown. And the reason for that is because Giotto, what it really needs as like the core starting point is just the cellular coordinates. And this is usually like your cell coordinates, the cell centroids, which this can be in 2D, the XY, or in 3D, XYZ. And in addition to that, you also need a matrix where you have like the cells on top and or the spots if it's not single cell resolution, and then the genes or some other type of features like proteins. In addition to that, you can also use your you can also integrate the tissue of interest that you have profiled. And so
Sorry, Ruben, can I interrupt for a second?
Right.
Your slides don't advance.
Oh, what about now?
Yep.
So now you see it?
Yep.
All right. So, okay. So I'm just gonna go back one slide. So Giotto needs, as I said, as input just the coordinates and the matrix, but you can also incorporate your image. And so what you have then, you create a Giotto object, which is using like create Giotto object. And by doing that, you can also integrate your Giotto image if you want to, so that you can overlay all the analysis on the actual image by just adding Giotto image. And there's a little bit more information here if you need it. In addition to that, for some of the functions, we also have, for example, for Visium 10x, we have some wrappers that where you can immediately just say create Giotto Visium object on the folder with all the information, and by doing that, you have the whole Giotto object. So the next couple of steps, what you can then do, and we will explain this in more detail in the next couple of slides, is you can do clustering to identify the different cell types that are available. Once you have done that, if you do not have single cell resolution data, but you have multiple cell resolution data, for example, the Visium data, you can also borrow information from single cell RNA-seq data sets or any other type of information to do some kind of like spatial enrichment to identify the cell type distribution. And then we have a number of different or novel tools to identify which kind of spatial patterns are present in your data set, and also like looking at cell neighborhood analysis. How is gene expression influenced by different composition in the cell neighborhood? And last but not least, once you have all these results, you can then incorporate them or upload them to our interactive visualization tool, the Giotto viewer, and visualize in an interactive manner. So now I'm just going to give the floor to Hupong, who will talk about the cell type identification. All right, thank you.
Thanks for the introduction, Ruben. So now I'm going to cover the cell type analysis part for Giotto. So the input for cell type analysis in Giotto is simply the gene expression matrix and the cell location matrix. And the purpose of this part is to learn the cell type information and also visualize the cell type information, both in the gene expression space and the physical space. So Giotto does that by a series of functions. Now I'm going to demonstrate the key steps in this procedure using a dataset generated by Murphy's technology on a piece of preoptic region in mouse. So in this dataset, overall 155 genes were profiled in a number of two-dimensional slices along the z directions. So to initialize the analysis, we take the expression matrix and the location matrix as input and use the create Giotto object function to initialize the analysis. Now we can already visualize the tissue architecture in the physical space. However, the internal information is not known yet. So afterwards, the next major step is to do the dimension reduction in the gene expression space. There are a few key actions was taken here. So the first is to use the filter Giotto function to filter the unreliable genes or cells from the input matrix, and then normalize the gene expression matrix. Afterwards, the key step here is to use the run PCA function to reduce the dimension of the gene expression of the cells in the gene expression space. And afterwards, we further reduce the dimension using the UMAP function to reduce it to two-dimensional or three-dimensional for the convenience of visualization in the following steps. Now we can visualize the data in the UMAP space. Now we can already see some of the structures in the data already. Some cells form clusters, and clusters are kind of far away from each other. Then the next step is to quantify this structural information we learned here. So Giotto does that by using a graph-based clustering approach implemented in two functions. The first one is to create a nearest neighbor network, then followed by latent clustering approaches. So after these two steps, we can visualize the cluster information in the gene expression space already. So the next question is, we also want to learn the biological identity of these different clusters. Giotto does that by using this find markers function to learn the cluster-specific markers. Here I'm showing the top markers identified for this particular data set. We can see some of the markers are expressed specifically for certain clusters, while some are not. So based on this marker expression pattern in different clusters, the user can then manually annotate the biological identity of each cluster using this annotate Giotto function. Now, this heat map summarizes the correspondence between the markers that we checked and the correspondence between these markers and to the annotated biological clusters of this data set. So now we have learned the cell type information in the data set. Then we can simply visualize the results both in the gene expression space and the physical space using this one function. Now, on the left-hand side, we can visualize the cell type information in the gene expression space. On the right-hand side, we can visualize the cell type information in the physical space. We can already see some important structural information in the physical space already. So for this particular three-dimensional data set, we can also visualize a particular two-dimensional slice for this as a subset of this data set. So Giotto offers functions to do that as well. So this background plot gives you a cell type information both in the physical space and the gene expression space. And also another function will give you an interested marker gene expression in the physical space and gene expression space as well. So if we look at the Murphish three-dimensional data set, it's kind of special because the X and Y direction is kind of continuous. However, for the Z direction, it's only discrete values was profiled. A potentially more general type of three-dimensional single-cell data set. For example, this is one data set that has been generated by StarMap technology. In this case, all of the three dimensions are continuous. So for this particular case, it is interesting. It will be interesting to look at the cell type or gene expression pattern for a user-specified virtual two-dimensional slices. So in Giotto, it is pretty straightforward to do that using a set of cross-section functions. So as you can see, you can first create the cross-section and also visualize the cell type pattern in a 2D plot as well. So similar things can be done for the gene expression pattern as well. So now I've covered how Giotto can do the cell type analysis for single cell technologies like Murphish and StarMap. As we all know, there are many non-single cell resolution technologies out there. So Giotto can also do cell type inference for those technologies. And this will be the topic for our next speaker, Ray.
Ray, are you ready?
Yeah. So
Ray, can you use this regular mode? So here we have the presenters mode.
can you see it?
It's still the presenter mode. Yep.
Sorry for the late. We can say that Giotto can not only deal with the single cell spatial expression data, but also Giotto can also deal with the lower resolution spatial expression dataset. And today I will focus on the cell type enrichment analysis for lower resolution spatial expression dataset. And we know in the single cell spatial expression dataset, such as Thickfish Plus and Morefish, we can do the clustering, and based on the marker gene expression of different cell type, we can annotate the clusters, and we can get the cell type information from the Thickfish Plus or Morefish or some other single cell spatial expression datasets. Recently, there are many lower resolution spatial expression datasets, such as VCM and SLIDE-6. They are not in the single cell RNA sequencing level. For example, this is the VCM mouse brain data. If we zoom this region, we can see that in one spot there are many black dots. So this indicates that in one spot there may have different cell types of this spot. So to overcome this challenge, we developed a different method to do the enrichment analysis for cell type in different regions. And here I just show you the page and the rank is the two different enrichment methods that could, based on the gene expression pattern of different cell types, it can predict the enrichment of a spot And the page and rank are two complementary algorithms. And for page, if you are interested in some genes and you know the marker genes or several cell types, and or you're interested in some cell types with marker genes, you can just using this marker gene list as the input of Giotto, and they do not need the expression value of these marker genes. And for the rank, if you have single cell RNA sequencing expression data and you have already labeled all the different cell types, you can use the single cell RNA sequencing expression and as well as the cell type labels as the input to the enrichment analysis. And both these two methods have implemented in the Giotto, and based on the Giotto enrichment, we can visualize the enrichment results. Here is a sample. And to further introduction, I will use the VCM mouse brain dataset from 10x. And here is the reference of coronal mouse brain from Allen Brain Atlas. And here we do the enrichment. Sorry, yeah, this figure seems too large. Yeah, here we use the marker genes from this year, the cell paper from Linus's lab, and we use the function implemented in Giotto create spatial enrich. And by using this function, we can calculate the enrichment score for each spot. And then if we want to visualize this, we can use the spatial plot. And here are the, we selected 10 cell types. And here is the visualization results. And I will further go some details into 4 specific cell types from the peptidic to the MSN. Also the granule neuron and astrocyte and epidermal. And for example, the granule neuron is a well-known cell type. And we can say that by using the Giotto, we can see granule neuron cell are enriched in this region, and it is highly consistent with the granule cell layer reported from the Allen Brain Atlas. This is the reference from Allen Brain Atlas. And also it is consistent with the marker genes expressed in granule neurons. And the peptidic astrocyte and also the MSN are also consistently with the reference annotation from the Allen Brain Atlas. So here is all the enrichment and analysis, and then I will hand back to Ruben for the Giotto spatial analysis.
All right. Okay. So I guess so far what we have seen is, first of all, how can we identify which cell types are present in the spatial data set? And then also how, or if you do not have single cell information, how can we do some kind of like spatial enrichment? But what more can we learn from the spatial information that is part in our data sets? So for that, we have a number of spatial analyses that are integrated in Giotto, and they are focused basically on 2 different things. One of them is looking at spatial patterns, and the other one is more looking at the cell neighborhood analysis and how gene expression changes within the cell neighborhood analysis. So I'm first going to focus on the spatial patterns, and there are really different layers of spatial patterns. You can look at individual genes, but you can also look at co-expression models of multiple genes that have a certain spatial pattern. In addition to that, this is continuous, but if you're also like interested in a more discrete spatial domains, we also have actually previously published a tool that can identify that. So to exemplify these 3 different analyses, I'm going to make use of the mouse kidney data set. This is this coronal section of the mouse kidney, where on the outside you have the cortex and inside you have the medulla. But if you just look at the original tissue figure, you cannot really say a lot about the anatomical structure. But for example, if you would just overlay it with the number of genes that are detected within each spot, because again, this is the VGM data set, so each spot can have multiple cell types, you can already see some kind of structure, but not really a lot. So what do we do to incorporate the spatial information in the different spatial data sets that we looked at? So we have 2 different ways. One of them is we can create a spatial grid, or we can create a spatial network. In a grid, like all the individual cells or spots that are within a grid, they kind of like are associated to each other. But a more like a fancier way, or a more like easier way, is perhaps looking at the spatial network. And so we use the spatial network as the beginning to do a lot of different spatial analysis. So in a very simplistic way, let's say that this is, so each dot here or each round is a cell, and you can create a spatial network. And Giotto has a lot of different ways to make it based on, you can select the number of Ks, based on the radial distance, a minimum number of neighbors, or also you can make this the law 9 network, which is kind of like very, very nice, since it just connects the nearest neighbors without actually skipping another neighbor to go to that one. So the first thing you can just do, and we incorporated a new method to do that, is to identify spatially coherent genes. So let's say you're looking at gene X is consistently high in neighboring cells, and we have a very fast and efficient way of identifying these spatially coherent genes. So in addition to this one, so this is the one that the novel algorithm that we integrated, BinSpect, we've also integrated a number of other tools, for example, transseq and spatial DE, which were previously published, and also another tool that we renamed Silhouette Rank, which was based on the paper of Chen Zhu in our lab that was published 2 years ago. So if you just look at the type of genes that you can identify, and this is just a small subset of some of these spatial genes, you can immediately see that there are very different spatial patterns that you can discriminate. But this is all just looking at. So one of the interesting things we looked at is how different are these different spatial tools, right, to identify spatial genes. And on average, what we see is that most of the genes are detected by all the different methods that you can see here in the middle, like all spatial genes, the very well-known spatial genes are usually detected by all different methods. Perhaps the bigger difference is in terms of speed. Let's, for example, if you look at spatial DE and transseq, which actually works very well, and they are very nice, and the design is very nice, but it takes them quite a long time. While if you, for example, see at Silhouette Rank, but also the BinSpect methods, they are much, much faster, which could be very important if you're looking at very big data sets. But let's say you're not really interested in an individual gene, but more at robust co-expression modules. You can also look at spatially co-expression modules. And what we mean with that is now you can look, for example, at gene X and gene Y, which do not necessarily have to be co-expressed in the same cell, but just in the neighborhood of each other. And so what we can do is we can just run this detect spatial correlation genes, and we can do this. And what you basically see is very similar as what you would do for another type of RNA-seq or single RNA-seq data set, where you look at spatial co-expression modules. So here in the y-axis and the x-axis are all genes. And so these blocks are spatially co-expressed modules. So how does it look like if you now overlay this again on the mouse kidney data set? It looks a little bit like something like this. So you can clearly see that there are these kind of spatial co-expression modules that are very distinct and that you can easily be extracted using Giotto. If you're more interested about now, okay, let's see this very nice ring structure, what type of genes are part of this co-expression module? You can, again, you can do that with Giotto, and you can identify all these genes that have these spatial co-expression patterns. All right. Then the last part, so this was all continuous. We also have this previous tool that was developed by Chen in our lab, which works on HMRF. And what it does, it doesn't only take into consideration the expression within your source cell, but it also looks at the expression in the neighboring cells, and it looks for shared spatial gene expression patterns. So by doing that, we can really transcend cell types and identify spatial domains, as you see here. And again, this is now illustrated again on this mouse coronal section, where you see this very discrete domains. So for the next part, this is the cell-cell interaction, which is more focused on cell types and how cell types influence gene expression. We will look at neighborhood composition. We will look at how neighboring cells might influence gene expression changes, and what can we learn about ligand receptor activity in terms of the spatial composition of your tissue. So for this one, we're going to use the SIGFISH Plus dataset from the somatosensory cortex. So basically, you're looking from the outer layers to the inner layers. And this is a really nice dataset because it has about 1,000 cells. But more importantly, each single cell has been profiled for 10,000 genes. So this is at least as good as a single cell RNA-seq dataset. So the first thing we obviously have to do, since this is a cell type specific analysis, we identify all the different cell types that are present. And as you can expect, since we're looking at the brain, there are these excitatory neurons, there are interneurons, you have these endothelial cells, all different kind of cell types that are somehow positioned along all these different fields of view. So the first very simple analysis you can do is just looking at neighborhood composition. Do some cells prefer to be next to other type of cells? So in order to do that, you can simply run the cell proximity enrichment, and what you get is either this heat map type of analysis or a network where you can just see if two pairs of cells are on average found closer in proximity to each other than what you would expect. And then if you, for example, would focus on one of them, like the oligodendrocytes and astrocytes, you can immediately go back to the spatial or the physical space and see where are these interacting cells. And then you can see that most of them are close here, which is probably close to the subventricular zone where these cell types originate. So that probably makes sense. And the next part is, let's say, how does gene expression changes according to the neighboring cell type composition? So for that one, you can use a find cell proximity genes function. So what it does is you're going to focus, for example, in this specific case, at endothelial cells, and whether these endothelial cells, when they're they're kind of scattered around the whole tissue, right? But some of them are close to astrocytes, others are close to inhibitory neurons, or close to this layer 4 excitatory neurons. And that might actually have an effect on the gene expression levels within these endothelial cells. For example, when we look at all these different endothelial cells, depending on whether they are located next to different neighboring cells, if you look at the endothelial markers, they don't really change a lot. So that means that there are indeed all endothelial cells, and that is not influenced by the neighborhood composition. But you can look at other genes that are influenced by the neighborhood composition. For example, those genes are being upregulated in those endothelial cells that are close to inhibitory neurons. And the other way around, it also works. So this is, for example, genes upregulated in endothelial cells close to layer 4 excitatory neurons. And sometimes you also see genes being downregulated. So this is just looking at one particular cell type, endothelial cells, in relationship to other cell types. You can also just visualize this for all the different combinations of cell types to cell types. So what we see here are these interaction change genes, as I just explained to you before, and how many of these interaction change genes do you see with these different cell type combinations. But so far, this is all focusing on one particular gene and how one particular gene changes, right? We can also think a little bit more about how do cells communicate with each other. And so we can look at ligand receptor activity. So in this very simple schematic, what we do here is we have all these different cell types coded by the colors, and they communicate using ligands and receptors. So what we can do just to get an idea is if they use a certain ligand and receptor, we can profile the expression of that ligand and receptor. So we're just looking at one particular ligand and receptor in this particular combination, for example, the green and the yellow that are actually in proximity. What we can then do is we can spatially permute them by just like repositioning all the yellow cells and all the green cells and see what is the expected distribution in the spatially permuted background. And based on this value, we have an idea about the spatial difference. So in this case, that would mean that if these two cells are in proximity, they seem to use this specific ligand and receptor more than you would expect by just a random chance. We can do this for all the different cell type combinations. And by doing that for one specific ligand and receptor, you know if a ligand or receptor is spatially more active or less active in which cell type combinations. So this is just visualizing this for one cell type, one ligand and receptor pair. We can also do this for multiple ligand and receptor pairs by just calling this spat cell-cell communication. So this is short for spatial cell-cell communication. What we see here is actually a subset of all the different ligand receptor pairs here in the y-axis, and here are the cell-cell pairs, so the cell type pairs in the x-axis. And if it's red, that means that this particular cell type pair uses this ligand receptor pair, a ligand receptor pair more than you would expect, and while blue is less than. So this was the last part for the spatial patterns and neighborhood analysis. So now for the last part of this webinar, we're going to look at how we can visualize all these results that we just explained to you in a very interactive manner. And this is going to be done by Jen.
Okay, thank you, Ruben. So let me share my screen. So my name's Chen, and so for my section, I'm going to talk about the interactive component. So specifically, we have built this application called Giotto Viewer that allows people to interact with the spatial genomics data. So what essentially is a Giotto Viewer? It is a locally hosted website that incorporates not only the gene expression, cell annotation information, but also information coming from images such as fluorescent images, cellular morphology. It brings all these data together in a multi-layer viewer. The input for this viewer could be a Giotto object exported to a set of specifically formatted files such as gene expression, annotation, and coordinates. And in addition to that, by adding the fluorescent images, including NISO, DAPI, or cell compartment staining, the Giotto viewer would display these information by overlaying the images as well as cell annotations all together. And by doing so, we can delineate cellular morphology. We can also color the cells by different color. We can also show the NISO staining as background. Okay. So this is how the Giotto viewer is organized. Essentially, by loading the Giotto, the data set first in Giotto viewer, you are presented with a canvas showing all the information that is out there. And you can use the plus and minus on the top right corner to zoom in or zoom out or move around by dragging the mouse. You can look at this section, which will allow you to select a particular staining, such as NISO DAPI. This will control the background images. And you can also, you can also see the red line outline here, which delineates the cellular morphology. By clicking on the cluster and the annotation, you can control what cell annotation you want to display. And in addition to that, you can click on these two buttons to specifically hand select any regions of cells you're interested in in order to export them for further analysis. And finally, you can use this box here to search for gene expression. And at the end, I will also talk about the transcript localization functions. What is unique about Giotto Viewer is that it allows people to put side by side multiple panels, which would display different properties of the data. So by doing this, you can actually get a good glimpse of how the cells are arranged in the physical space as well as in the expression space, such as the t-SNE or UMAP. So what I'm doing here is that I invoked 4 panels here. Two of the panels showing physical space and 2 of the panels showing the arrangement of the cells in the t-SNE. And here, this is the somatosensory cortex data from SeqFish Plus, which gives you very rich information of 10,000 genes, and as well as the NISO DAPI images, along with cell segmentation information. And by putting these panels side by side, you can appreciate the fact that some of the cells have different annotations. For example, cells in the same spatial domains are composed of various different cell types, such as indicated here in layer 1, and all the different cell types indicated here. And you can also highlight any one of these cells, and immediately the corresponding cell in the gene expression space will also be highlighted as well. So you can actually see where these cells are located in the expression clustering. And Giotto Viewer does not work only for single cell resolution data. It can also be used for the 10x genomics platform. So here I'm using one of the test data set provided by Visium, which shows the mouse adult brain coronal slice. And here, this is a demo showing first the ability to highlight specific cluster. And you can see immediately in the other panel, the cluster is highlighted. You can use the toggle function, which I've started here, to hand select a region, as I've done here. And by doing so, you can also see the corresponding cells in the expression panel. And next, the zooming function is really great because it allows you to see what is underneath each spot within the 10x Visium. And you can further fine-tune the cell selection. So after doing that, you can click on that population of the cells to view the cell ID or save the cell selection for further analysis in Giotto Analyzer. And Ray earlier talked about cell type enrichment result, which gives us a very continuous representation of the data so you can actually show the cell type enrichment for for example, oligodendrocyte as indicated here, or peptidergic neurons as indicated here, or the cortex hippocampus region, which can all be toggled within the annotation section here. And finally, this is the medium spiny neuron sections of the brain. So all of these annotations will be automatically exported when you invoke the export Giotto functions. And last, the Giotto viewer can also be used to visualize subcellular information data. So if you, so for technologies such as CFISH Plus or MERFISH, these usually provide, you know, single transcript location within these cells. So here, the individual dots indicate transcripts within these cells. And by zooming in or out, you can actually appreciate where does the transcript located inside the cell? Is it in the nucleus or in the cell periphery? So here I have tried to display 3 genes of interest, GJA1, SLC10A7, of the different cell type markers. You can, in combinations with the cell type annotation, which is indicated by the color of these cells, you can actually see what is the cell identity as well as the transcript localization. And you can click the All Genes button here if you want to just show all the transcripts inside the cell. And that could be useful, for example, for cell segmentation purpose. So by now I have provided some you know, demo of what Giotto Viewer can do. And we think that Giotto Viewer will provide a good supplementary website for future spatial expression projects. And if you're interested in setting Giotto Viewer up, you can visit the links provided here for details about setting up a data set or getting more details about the usage. And lastly, just to summarize everything that we have talked about here, we have created a website containing all the details of today's presentations in spatialgiotto.com. And more instructions about individual components of Giotto are provided by this development link here. And also a paper mentioning today's work is deposited in bioRxiv. So this is the website that was introduced. So and give you some of the idea here. We can also set up, get information about, for example, Giotto Viewer setup. And all these slides, by the way, will be posted on the website. So these are going through some of the steps. So that's all I have for now.
Okay, so that concludes the presentations of the day. If you have questions, you can ask by raising your hand or by typing your questions in the chat room. And you know, if you raise your hand, you can talk. I can unmute you. So oh, okay. Yeah, because you can talk.
Hi, yeah. My name is Nicholas. I am from the Elowitz and do collaboration with Long's lab.
Hi.
So for one of the projects that I've been helping a little bit, that has to do with subcellular, so looking at patterns of subcellular localization of the RNAs, looking at, for example, where the distributions are random or non-random within individual cells. Have you already created analysis tools for that? Or maybe analysis tools that have been thought for the level of the tissue that could be applied, you know, it will be relatively easy to re-implement the same algorithm for single cell analysis of the distributions of the transcripts.
So at this point, the co-localization analysis is still done at the cellular level, at the tissue level. We are still very interested in this cellular co-localization functionality, so that's still under investigation. So yeah, so have to check out later.
Can I ask another question?
Sure.
what language is the program written in for the tools, the different modules of the tool written in?
Okay, so the main language, I will refer this to Ruben, so he will give you a better description than me.
Oh, sorry. Can you repeat your question? I was
what language? So you have different modules. Are the different modules written in the same language? What language was used to write all the tools?
So most of it is actually written in R, and there's also a lot of other like specific, more specific analysis written in Python. And what we make heavily use of, if you're more familiar with that, is the reticulate package, which you can easily integrate Python and R.
And is it easy, for example, to implement or to call it? Say, if you're, most people nowadays, I don't know if most people, but a lot of people are using Jupyter notebooks to sort of like do, you know, analysis and publish that as a, you know, sort of like set of notes of that reproduce the exact codes that reproduce your figures in your paper. Is it easy to say run this through a Python sort of like Jupyter type of notebook, even if it's been run by, say, an R code?
Yes. So you can actually run the code in a Jupyter notebook. We actually have been thinking, but we didn't have enough time to put this on, what's it called again? Google Colaboratory, which is this online notebook. So we have been thinking about also putting our examples there, but we don't have enough hands to do everything.
It's a very good job. I really like what I've seen. Thank you so much for creating it. It's going to be very useful.
Thank you very much. So I think there is a question in the chat room. Maybe that's already answered already. So I think there's a question saying that does Giotto provide a standard single cell QC metrics, like a number of spots per cell, unique genes per cell, etc.? Maybe Ruben can say something about that?
Yes, we actually have a number of functions that do these kind of things, that look at how many genes can be detected per spot, per cell, like what is the percentage of, for example, mitochondrial genes, if you have genome-wide data, of ribosomal genes, and lots of other, like whatever you do in a standard single-cell RNA-seq pipeline can also be applied with Giotto on a spatial data set.
So there's a question about if it is possible to incorporate the multiple slides or sections in the future implementations. I think I can say something briefly, and maybe Ruben and Chen could expand on that. I think right now, yes, the short answer is yes, you can. So if you have information from multiple slides, we have the function to stitch different fields together and analyze these together. Treat as the bigger field. I'm not sure if Ruben or Chen has something else to add.
Yeah, so just a SeqFish Plus example data set gives an example about how to do that. It does require some kind of like manual stitching, so you need to know a little bit like where the different fields of view were coming from, but it is possible.
I guess this is a question for you, Ruben, again. You asked too many questions. So can we have a list of the neighboring cells and their distance based on the chosen spatial network? I guess the short answer is yes, right?
Yes, so I replied to that question already.
Oh, you did already. Okay, sorry, I missed that. If you add a question or results from your own analysis, that's interesting. So yeah, I mean, like, can you add the results from your own analysis and add to this data structure? So so for one thing, it's so the short answer is yes. So Ruben is nodding, and you can also use that information. You can visualize that as well, right? So remember that we have two modules, both the visualization and also the analyzer. So maybe I don't know if Ruben and Chen has anything else to add.
Yeah, so I think it's possible for both, and I think Chen can talk about the Giotto viewer part. But for Giotto analyzer, if you already have some kind of information for, let's say, the nearest neighbor network or some other type of network. That can all be given when you create a Giotto object. So if you have another tool or whatever you would like to use, but you then would like to use Giotto to do some of the more downstream analysis, that's definitely possible.
So for the viewer, the input can be user-specified cell annotations. So you can actually run in different ways. So in the most simple way, you actually don't even need to have cell annotation. You can just browse the cells and use it as to search for different gene expression, for example. So we have sometimes some user requesting that. But if you have cell annotation, you just need to format it in a specific way to use a Giotto viewer.
Anybody can comment about this initial normalization?
Yeah, I can also comment on that one if you want. So the initial normalization, that actually depends a little bit on the type of data set you have. But for example, for SIG++, and it's so similar to single-cell RNA-seq, we do what most people do, and that's just you normalize the library size, and then you log scale your values. And that seems to work really well. But in addition to that, so if you have a different type of data set which has their own typical problems, what you could also just do is have your own type of normalization and just give that to the Giotto object. So that also works. So if you think like Giotto does not have everything that I need to normalize my data properly, you can kind of like circumvent that.
So somebody asked about the segmentation. So the Giotto right now does not have segmentation functionality. So I mean, this is a really difficult task, as you may imagine. It's highly platform specific. So, but if you have your own, segmentation using, I mean, there are a lot of softwares out there available, then we can take that information and analyze from there. So if you want to do this, directly use the things that are already existing in the analyzer, then you would need to convert that into the cell by give the location, right, a coordinates for that cell. That's the way it is right now. So for example, if you have a very complicated morphology, then you would need to map the distance. I mean, right now we don't have the ability to account for the complex morphology yet right now. But if you have multiple fields with the images, as I mentioned before, that's not a problem. We have internally, we have a functionality to stage these together, so you treat that as a single big field to do the analysis. I should also mention that perhaps you already noticed that we can do this not just 2D analyze 2D data, but also 3D data, right? So it doesn't have to be like a 2D field of view. The same idea, you can stage this together. And for 3D data, we can also generate this, the virtual slices, right? So it's maybe different from the imaging field is taken. How would the normalization affect the clustering results? Yeah. I guess, I mean, it's the same thing as the single cell RNA-seq, right? So, I mean, it's there's a standard the normalization methods that are tested to be, robust, right, with the various these clustering methods. So that's nothing unique about the spatial transitomic field. So everything, every experience you have from the RNA-seq data should also work over here as well. I don't see any other questions in the chat room, or anyone want to ask a question still? I don't see.
Yeah, so there's one question about Giotto visualization handling large 500 megabyte pixel stitch images. Actually, our images, when they are stitched, they're actually very, very large. So we have been handling data, for example, from 10, 20s, or even 30 different fields. But it is generally not a challenge because the way Giotto Viewer works is that it splits up the large images into individual tiles and use this kind of like tiling-based, Google Map-based approach to visualize the data at different various zoom levels. So by doing so, it significantly cuts down the information that needs to be displayed at once on the screen, and that's how it handles very large data sets.
Okay.
There's also a question about if the spatial grid is only a visualization tool, or can we also use these grids for further analysis? So the answer is that you can also use it for further analysis in a very similar manner as you would do with the spatial networks. For example, in the co-expression modules, but also to do some kind of like spatial PCA. The reason we don't do it is because right now we seem to favor the spatial network over the spatial grid.